in every discipline, there are specific tools that are used very frequently to help people understand and communicate observations, data, and conclusions. So if we were to look at any lab in a cell biology discipline, the primary tool that we would see that they were using is the scanning electron microscope. If we were to go to physics and see within that discipline, we would see that they were using a combination of tools, primarily a lot of computer modeling. And so within every discipline, we know that there are specific tools that are used frequently, and evolutionary biology is no different. In evolutionary biology, we are going to be using phylogenetic trees, and this whole chapter on tree thinking is really helping us to get used to using and reading this abstract tool to show the relatedness between species. So it's very powerful. We can communicate quite a bit of information very quickly, and that's part of the reason why we love it. We love things that we can quickly communicate a lot of information. Now, behind the scenes, there is a lot of background information. There's a lot of evidence and data that goes behind the abstract image that often looks fairly simple. So some of the things that we're going to see, that data behind the scenes, will be DNA genome comparisons, protein comparisons, so looking at the whole proteome of a species and comparing it to another species. Increasingly, we're going to see even other molecules. So molecules that are affecting gene switches, turning them on and turning them off, affecting gene expression. We'll see some of that evidence behind a phylogenetic tree. And of course, we're going to see morphology, those physical characteristics, whether that's the skeleton, the muscle structure, the implied muscle structure, or whether that's even exterior things, like looking at something like a feather. And I particularly enjoy the research about feathers and their development from scales, that connection between birds and lizards going all the way back to the dinosaurs. That is quite a rabbit hole to be able to fall down. So if we are dealing with phylogenetic trees, though they may look very simple, please remember that there's a tremendous amount of data behind the scenes. The other thing that we have to assume we're looking at a phylogenetic tree, another underlying piece of evidence, is what you see in the beginning of Dr. David Baum's Tree Thinking article, where you've got multiple generations and you're modeling and showing how genes are being inherited, how they're being passed on to the next generation, and then you're braiding and you're linking all of those shared lineages together into that shared line. So all of that's there behind the scenes. So when I draw an image like this, it's fairly simple. I know that I'm drawing a small portion of the universal tree of life. And so one of the important things in my phylogenetic tree is going to be my root. This little line here is implying and communicating that I know that this group of species that I'm interested in is connected to all the rest of the life on this planet. But I just want to zoom in on this one small portion of that universal tree of life. So here I'm going to have several different species. And one of the things that we'll notice is that there's lots of ways that I could show the relatedness, the relationships between these species. I personally like this form of a phylogenetic tree. To my eyeballs, it makes a lot of sense. But you'll often see that these phylogenetic trees can be turned on their side and have sort of a rectangular shape. And then there's the circular phylogenetic trees. If you remember back in an earlier set of lecture slides, we had the Canidae, all the dogs, on a phylogenetic tree, which was that beautiful circle. Visually, it looks stunning, but for my brain to be able to trace which species are closely related to others, to clearly identify those nodes, I find the circular version incredibly, incredibly challenging. So you'll have preferences. You'll have phylogenetic trees that when you see them, you instantly get it. And other versions of phylogenetic trees other orientations that just make your brain work harder and don't work for you. Everybody seems to be a little bit different. This is why I show you so many different types of phylogenetic trees, is because I need to find out what works for you, and you need to find out what works for you, how your brain is intuitively reading and understanding these in order to understand the relatedness between the species. So we've got our root, implying connection back to all the rest of life on this planet. And then each of our branches here are connecting back through these nodes. And these nodes are implying some kind of speciation event. Some kind of evolutionary event has occurred that is separating the species out here at the end of the branch from the rest of the species in this clade, in this group that I am looking at. So our nodes are incredibly important. They imply a lot of information. And so when I look at this phylogenetic tree, the way how I'm going to read this is that species A and B are closely related. They share one node in common. Species A is very distantly related from species E because we have 4 separate nodes, 4 evolutionary events separating species E from species A. Now, one of the things I might do is I might have included species E here as my outgroup. The reason why I would do that is because this is the last most recent common ancestor that has traits and characteristics in common with all the rest of the organisms in my phylogenetic tree. So they're going to anchor me. They're going to be my sort of baseline. If I were using the terminology of the scientific method, I might call this my control group. So I'm going to be comparing all the rest of the organisms back to species E, my outgroup. So I have an evolutionary event that has separated species D, a second evolutionary event that has separated species C, and then a third evolutionary event that separates both species B and species A. And one of the things I love about this abstract tool is that I can switch my species names. So if I switch species A and B here, I haven't actually communicated any new information. They're still closely related to each other. They're still sharing that node. But if I switch species B and D, well, now I'm communicating something significantly different. Now I'm saying species D is more closely related to species A. They now share a node in common. But A has 3 separate nodes separating it from species B. So do be careful when you are reading phylogenetic trees. Keep your eye open on those nodes. Those nodes are going to help communicate relatedness. Now, one of the reasons why we will use a phylogenetic tree or why we want to zoom in on a clade, a group of individuals like this, is because maybe we're looking for something like ancestral characteristics. What is a trait that species E has that sticks around even though we have all of these separate speciation events? So let's take for granted something like a spine. Species E has a spine. And once we see that trait developed, every species after that has that trait, has the spine. So what we would then predict or what we would then imply is that if we have future speciation events, we have a lot more other individuals that are new to this planet, new species, that are closely related to these individuals, that they would still have that ancestral characteristic, they would still have something like a spine. The other thing that we'll be looking for, the other thing we want to keep track of, are those derived characteristics, the new traits, the new adaptations. So let's say species C up here has a new trait. That new trait allows it to be able to survive in Arctic waters, waters that are very high in oxygen but are extremely cold. Maybe that new trait is the mutation in a gene, that repetition, allowing the gene to make an antifreeze protein that keeps the blood from freezing. Hopefully all this is sounding familiar and you're going, ice fish, right, exactly. What if species C has that unique adaptation? We would anticipate that species B and species A would also have that unique adaptation. They may not, though. That would be the interesting thing to investigate and to find out. So a derived characteristic is something that's new to a species. We haven't seen it before. It gives that species some kind of advantage, some kind of hopefully increase in fitness in response to the environment. But sometimes a derived characteristic is just a thing. If we have we're looking at something like birds, and we've got a bird that has a derived characteristic where the feathers on its head stick out at weird angles. Well, maybe that doesn't actually do anything for the bird. Maybe it doesn't increase its reproductive success. It doesn't help with its fitness. It's just an interesting thing. Sometimes we have derived characteristics that are like that. Not very often, but I didn't want you to come away with the misconception that every time a species is developing a trait or developing an adaptation, that's necessarily directly affecting the fitness. Sometimes it's just a thing. Sometimes we find out later that it does or doesn't affect the fitness, but for a while we just have to say, well, there's a thing, and that's cool. That separates these species away from the other species. So with our phylogenetic tree, sometimes we will do things like focus in even more detail. So maybe I just need to focus in on these 3 species. By cutting my phylogenetic tree here and preserving the relatedness between all of the species on this branch, I have a monophyletic clade. Mono being 1. I've made 1 cut to preserve the relatedness between the species. Maybe species A out there is nice, but I don't really care about it. I don't like it. I'm going to cut off species A, but I'm really interested in species B, C, and D. Now I have a polyphyletic clade. The reason why is because I had to make 2 cuts. I had to eliminate species on either side in order to zoom in on just these 3 individuals. Now the final thing that we will see sometimes, especially when we have new species or we're just beginning to work out the relatedness between the species, is we'll see a main branch like we've seen before. We'll still see the root connecting back to the universal tree of life. But maybe what I would see is I would see something like 3 individual species connected back to the same node. Now this looks really weird. It's something called a polytony. Poly being many. So I have many species connecting back to one shared node. What I am communicating when I do this is I'm communicating that I know that these are separate, distinct species. I have no doubt in my head that these are not all the same species, so I can't group them together with just one shared line. But the difficulty or the problem is that I don't have enough evidence to support the relatedness. So I don't know if C is more closely related to B. I don't know if B is more closely related to A, or how all of this is going to work out. I need more evidence to show the evolutionary events, to show the speciation that has occurred. So until I get that evidence, I'm going to collapse everything back to the shared node, because this I'm confident about. I know we've got an evolutionary event that has happened here. I'm just not sure about how everything else is going to work out. So you might see that sometimes with phylogenetic trees, this collapsing back to one shared node until we get more data and more evidence to help support the relatedness between different species. So most of the time in evolutionary biology, we want to be making, we want to be using, we want to be focusing in on phylogenetic trees. We want a very narrow focus. Now, when we step away from research in evolutionary biology, and we're looking at education, or we're trying to communicate larger concepts and larger themes, this is where we tend to see the use of cladograms and cladistics. Now with cladistics, what we're going to do is we're going to take really broad categories of species. So we'll have a category of something like trees, a category of grasses, we'll have a category of forbs, those are flowering plants, and maybe we just want to track, what are the differences, what are the similarities. We want to track these sort of larger scale patterns. Maybe we have a good reason for that. So when we get into doing cladistics in order to make a phylogenetic tree, we'll often make a data matrix. On the lecture slides, I tried to show you some different versions of data matrices. Sometimes what people will do is they'll put in X if a characteristic is present in a group of individual species, or they'll put a 0 if it's absent. Other people will use a rating system, a 1, a 2, a 3, to communicate, is that trait present but small? Is that trait present but bigger in this group of species? There's a lot of different ways that we see the matrices. For your cladogram homework, what you're going to be doing is using that binary system. If it's got it, it gets the number 1. If it doesn't have it, it gets the 0. Then you total up in each column, and based on those totals, you're able to make a phylogenetic tree showing the relatedness of species. So we really like data matrices for making phylogenetic trees. It takes away some of the ambiguity and some of the biases. We make decisions about the traits we're going to include. We take those measurements, present, 1, absent, 0, total it up. Here's your relatedness of the phylogenetic tree. So we'll get some practice with the cladogram homework, and I don't know how you feel about barnacles and lobsters and crabs, but it's pretty easy to tell ancestral versus derived characteristics for those groups of individual species. So that's why we're going to focus in on that. In the second half of the semester, we're going to take measurements on skulls from different species in the hominin family tree. And then based on your data, present, 1, absent, 0, based on your data, you are going to have a phylogenetic tree based on your measurements. So we'll come back and revisit this in Unit 8. We're looking at human evolution. So we've got our data matrix, and our data matrix is going to inform our phylogenetic tree. And just to follow through with my little example of grasses, forbs, and trees, the big question is, why in the world would I care? Why would I broaden my perspective to look at these broad categories? Which brings me back to the concept of a morphological species. Remember, we'll use that morphological species when we're trying to make comparisons in broad categories. So maybe I want to define a forest and a prairie. A prairie is going to have grasses and forbs, and my forest is going to have trees. Maybe what I'm looking at is I'm looking at different ways to manage this environment. I know that both prairies and forests need fire, and fire is very helpful to maintaining the health of those ecosystems. But the difficulty or the challenge is that fire is going to affect my prairie very differently than my forest. I'm going to be looking for different temperatures, how long I let it burn, the conditions with which I will be doing the burn, and then ultimately, what is my result? What am I trying to get at with this management technique? And so by using a morphological species category, and then using a cladogram with these broad categories, this could help me inform or help me communicate different management strategies based on traits and characteristics that these broad groups have and don't have, and how that might have changed over time. So this is one way that this can be really helpful. Of course, in the field of education, we rely heavily on cladograms because most of the time we're not diving into the details of individual species. We really are just staying at this large category level. So hopefully this helps you understand a little bit more about how and why we use phylogenetic trees, how and why we would use something like cladistics and cladograms. There's another tool that we talk about, the quantitative trait loci, and this is going to help us to identify genes that are leading to characteristics that we're interested in, and it relies on this concept of linked genes. So get to know the definition. We won't talk about it much in this class. I just wanted to expose you to this other tool in case you're going further in genetics or further in wildlife biology, plant biology, or cellular biology. We tend to see that concept comes up in those disciplines, even more so than it does in evolutionary biology lately, but maybe that could change. The last concept to touch base with is the concept of a living fossil. Remember, a living fossil is very intriguing. It's an organism, a species that hasn't changed much, even though we've got fossil evidence showing us how long this species has been on our planet. And I love these species. It's so amazing that despite the ice ages and the temperature swings and new predators and new species and new disease agents, new bacteria and viruses and fungi and all of the things that could try and kill you, that these species remain the same. Their morphology often stays the same. Their genetics are often the same. Yet we know so much has changed around them. They haven't necessarily given rise to lots of new species either. They're just kind of out there on their own. So a couple of great examples that we have. We've got the nautilus. Please watch that David Attenborough video. I think he does a fantastic job of narrating through the weirdness of a nautilus. And once you learn a little bit more about how it behaves, the question is, why isn't it dead? Why haven't these gone extinct, given all of the predation that we have in the oceans? So read the Into the Jungle chapter on the coelacanth. That discovery was very, very helpful. The morphology then maps onto cladograms and phylogenetic trees, helping us understand the connection of life on land to life in water. Though we don't cover it much here in evolutionary biology, when it comes to land plants, we've got a very clear understanding of how plants went from living in the water, where they had already dominated, to moving onto land, and all of those derived characteristics that they needed to be on land, including being able to tolerate the harsh UV radiation that damages DNA, being able to deal with the lack of water. And then, of course, why would you come up here on land if you're a plant? Tons of food, sunshine, and absolutely no competition for those first plants. So we tend to have a better understanding when it comes to plants moving onto land than we do about animals. But this is why both coelacanth and Tiktaalik, that's mentioned in the textbook chapter, are such crucial discoveries, because they're helping us really deepen our understanding of how animal life went from life in the water to life on land. As always, if you have any questions, please make sure to email me or drop into Zoom office hours.